But I would like us to get right on our topic for today, and our guest for today, Alexandros Makryannis, and we are very honored to have him with us. Professor Makryannis is the director of the Center for Drug Discovery here at Northeastern University. He specializes in drug design synthesis, molecular recognition, membrane biophysics, membrane protein purification and characterization, nuclear magnetic resonance spectroscopy, and high-throughput technology, all subjects with which I have great familiarity. Okay. At the center, they study the effects of drug molecules on membranes using solution and solid-state nuclear magnetic resonance and computational methods, and use the results, and the results are used to design and synthesize improved medications. Now, I can't stress enough how important this kind of work is, and how insightful I think it will be for us to look at it through the lens of design. So with that, let me welcome Dr. Makryannis. So you can just control the slides. Yes, it is. What did I just do? Just a second. Nothing to worry about. There you go. So
okay, and this is this way.
Okay,
and I'm on.
You're on.
Yeah. Okay. Am I on?
No, it's not.
Oh,
it's not for broadcast. It's
okay.
Just for recording.
All right. So hi. Well, you've had talks about design of buildings and other things. So I'm going to take you to a lower level of size. Okay? So it's going to be designing molecules. You can't see the molecules, but they're there, and they have a shape. They even have colors on them. If you want to do some colorful designation of their properties. And drug design is designing molecules that do good things for humans and animals. And the principle of drug design is to get the right shape. I'll explain. So think about a large molecule, which is part of your body, DNA, an enzyme, you know what an enzyme is, or a GPCR, G protein coupled receptor. They're molecules that have a molecular weight of 50,000. And then the drug is a small molecule, has a molecular weight maybe 300, 400, 500, 600. Okay? And you have the protein, which is in this case the enzyme, functions in the body and has a special role. So what we want to do with the drug is tweak that enzyme, because something has gone wrong in the body, and we want to fix it by tweaking one of these proteins. So that's what it's about. Here we are. So you see, yeah, okay. So you see here what the enzyme is, and it has a particular place for the substrate to fit in. And the substrate could be another molecule that's present in the body. Let's say if the enzyme is digestive enzyme, the substrate may be a sugar, a disaccharide that gets chopped into two. If the substrate is some sort of a modulator of brain function, then the substrate goes in there. Now, what we do then is we take a drug that we have designed. That drug then fits the same place where the substrate is. So when the substrate interacts with the enzyme, it forms a complex See, it gets in here, forms a complex, and it gives you a product. Let's say you take sucrose. Sucrose is made of 2 sugars, and the product is glucose and fructose. That's the product. Now, I make a drug. The drug fits in here. And now this enzyme cannot split the sugar anymore because that drug is sitting in there. It prevents it from. It inhibits the function of the enzyme. It's called an enzyme inhibitor. It's an enzyme-drug complex, no product. So if your body is producing a lot of something and you want to diminish the amount of what the body is producing, you get a drug, and the drug tells you, slow down. So the question is, how do we design this drug? Let me take you to a different scenario. So this particular target is an enzyme, and enzymes come in different shapes and forms, and they can be water-soluble or they can be membrane enzymes. They can have a molecular weight of 20,000, sometimes 15,000. It can go as high as 200,000, 300,000, large molecules. Now, these are generally, where I portrayed it here, molecules that can sit inside the cell, and they're soluble inside the cell. Alternatively, we can have this sort of a signaling receptor. Now, that receptor now is again a protein, and think about it as a switch. Okay? So when the receptor is turned on, you can feel good. If it's turned off, you don't feel anything. If the receptor is turned on, your heart beats fast. If it's turned off, it stays the way it is. So there is a hormone in the body. For example, you've heard of adrenaline, or acetylcholine, or dopamine. Anyone who does biology here? All right, so, okay. So dopamine, and then this hormone then gets into the receptor, fits in there, forms a hormone receptor complex, and as a result of this, a whole set of activities happen beyond there. So this is the initiation of the activity. I'm not going to talk about what activities happen, but at the end of the day, let's say this one, when it fits in, you could get a increase in your body temperature. That's one effect. Or you could start laughing. Okay, so that's the end point. So if this now is a situation where this particular switch is not well adjusted, you have high fever, you want to bring it down, then you want to do something with that receptor. So we make a drug. The drug fits in here. It prevents the hormone from coming in here, and it doesn't give you the effect. So that's a general story about how drugs work. And the question is, how can we design this drug? We want to make sure that it fits well, first of all. So it's all about fitting. And that's what this design is all about. How you can make this molecule, and we're talking now of a small molecule, molecular weight 3, 4, 5, 600, fitting in a larger molecule. And when it goes in there, it prevents this large molecule from doing its job. It becomes an inhibitor. So it's an enzyme inhibitor. And if it fits in that receptor, it becomes a receptor blocker. And you all heard about beta blockers. People who have high blood pressure, they take beta blockers. That's what the beta blocker does. Goes in here, prevents the hormone, which in this case happens to be epinephrine. And epinephrine increases your blood pressure. You put the drug in there, it prevents that increase, brings your blood pressure down. So people who have high blood pressure take beta blockers. So how do we design these beta blockers? Okay. Now, I wanted to also acquaint you with another possibility, because this thing can become more complex. So I talked to you about having a ligand or a drug get in here, forms a protein ligand complex. However, there's another way you can tackle this. A drug can come in here and modify this particular crevice, which is called the active site, and then allow the normal hormone to come in. So the drug then doesn't go in the active site, which is called the orthosteric site, but it goes in a different site. It influences the shape of the active site, and you get an effect. This is called an allosteric interaction. So the point I wanted to make is there are many ways of modifying the function of a protein, and it can be done either by tweaking it directly where its function is or by going around it. Now, how do we do this? Let me talk to you about something we have done in the lab. All right? This is an enzyme here. That's the structure of the enzyme. All of these are peptide bonds. This is called an alpha helix. So there are alpha helical arrangements. This is called a beta sheet. You see, all these are beta sheets. These are alpha helices. And that's the shape that proteins work, because the protein is made up of amino acids. And these amino acids, they interact with each other. And the message is that at the end of all these interactions, this protein must have a particular shape. And this is the shape of this protein. It has a molecular weight of about 36,000 molecular weight. It's called human monoacyl glycerolipase. And we've shown that this is the structure of it. How did we show it? I'll tell you about that. So now we've decided that we need to know then, if we're going to make the drug correctly, we need to know the shape of the protein, and we need to know the shape of what the drug should be. So we study both. We study the shape of the proteins, and then we design the drugs so they can fit where they need to fit. Okay. How do we find the structure of the protein? Well, let's see. So this is the E protein here. Then we zoom in on where the business side of the protein is. What does this protein do? The enzyme catalyzer reaction? It all happens in here. So if you magnify this piece here, you'll see that it is made up of this particular crevice, and there are specific amino acid residues that participate in this activity. There is an aspartic acid here, a histidine here, and a serine. These are all amino acid residues. And this is called the active site. How do we learn about this? How do we get the structure? There are several ways. These are the tools that we use to look at the structure and the shape of this protein. So one tool we use is called the nuclear magnetic resonance spectroscopy. So we made that protein. We actually cloned it. And then after we cloned it, we had the bugs produce it to express it. And then they made that protein, and we purified that protein, and we put it in a tube. And the tube called NMR tube, nuclear magnetic resonance, it gives us a spectrum. Actually, it gives us many types of spectra, but this is one of the spectra we have. And we use this kind of information to deduce and calculate the structure of the protein. So now we have used nuclear magnetic resonance, or NMR. NMR, you probably heard about it. It's same technology as MRI, except done in a different way. Actually, NMR came before MRI. And MRI was invented through the NMR method. The NMR tells you a lot of information about each atom, how these atoms interact with each other. So that's one technology to use. We can also use something called crystal structure. We can crystallize this enzyme, and then we find out what the structure is. And a third way is to calculate it from scratch. Then you do computational. We do two of these. We do the computational part and we do the NMR. And with regard to crystallization, we collaborate with other laboratories. So now we have some idea about how we deduce the structure of the protein. Then we sit down on a piece of paper, on the computer, etc., and we try to make molecules that would fit in that place. Because what we want to do now, see these three residues, they zoom in the molecule we put in, and they have to do something to it. In this case, they have to split it in two, and they do that catalytic activity. We want to prevent it from doing this. So we want to put something that fits in here but doesn't do the job. That's called an enzyme inhibitor. And we can do that by using the information we have here. We could use the computer. We could actually use a piece of paper and design things on there. I'll show you. So this is the NMR. And then we designed these two molecules here. Actually, this molecule here, AM6580. There it is. And it fits right in here. So how did we design this? Well, as I said, in the same way we've gotten the information on the protein itself, now we get the information on the small molecule. And sure enough, it seems to work, because when we test this enzyme, this molecule, the drug, AM6580, which we hope would be an analgesic compound. In other words, if you have intense pain, you take this compound, it stops the pain. And the way it stops the pain is by stopping the function of this enzyme. Now you see it here. Okay? Actually, what this enzyme does, give you a little more detail. So this enzyme, see this molecule here, it comes and one of these amino acids interacts here and cleaves this bond. It cuts it in two. And this piece ends up fitting in the enzyme, and then, as you've seen in the cartoon, the substrate cannot get in. So it inactivates the enzyme. It's an enzyme inhibitor. Now, how do we end up designing these molecules? Ultimately, we want to design them for a perfect fit. So we have to play around with this. We have to look at the shapes of these molecules. Maybe we want to tweak it a little bit here to make it to fit a little better. Just like you go to the tailor shop and they make a dress for you. It says, well, I need to cut it here a little bit. And we are the chemists. So we go in, and how do we cut a piece from the molecule? We just go and cook another molecule which has a smaller one. Because the way we make these is by cooking. We get these ingredients and we build them up, and that's how we made. This is a molecule which we have constructed in our lab. We have a student who comes in, and they run several reactions. We make this one molecule, and it fits in here. Now, we want to take advantage of the information we got on the structure of the enzyme, especially the structure of the place where the hormone or the substrate fits. So we design the contours of this, so we can design molecules that can fit into the contour, and we call it a pharmacophore model. Let me explain. Here we have. So this is a piece of the enzyme, and we know that if the molecule needs to fit in there, it has to have this kind of shape. So any molecule we do must fit in here. So we can then we already have information how it fits. Not only does it have to fit there, but we want to make sure that it doesn't fit anywhere else. Because you can't make a molecule that fits everywhere. It creates havoc. Imagine, you're laughing and getting a high temperature, and you're doing all of these things happening together. So we have to make it very specific. So that's called selectivity. And the big competitor here in our design is another enzyme, which is called fatty acid amide hydrolase. So we want to make molecules that fit only here, but not here. So to make sure they don't fit there, we also work on the other molecule, and we've constructed this pharmacophore model. So the molecules that fit here should not fit there. That's the kind of games we play. That's called a pharmacophore model for designing enzyme inhibitors. Okay? Now we play a little more games here. So here we magnified this. This is all, by the way, computationally determined. In other words, we got the structure from the NMR, etc., put them on the computer, etc., and we zoom in. We have like a magnifying glass, and we know this is where this thing has to fit. And there's the molecule. Our molecule fits right here. You see? AM6580. Now, what happens when it gets in the enzyme? As I said, this enzyme likes to do some chemistry on it. It chops it in. So let's see how it does that. So we know that this fit, but there is more here. So if we wanted to tinker around, we make a molecule that goes maybe a little more here. So we're designing it to go more in there, because there's room in there. And there is room here. So we know now where we can go and modify. The whole idea is to make it fit the best way possible. And we have ways to measure how tightly it fits. Parameters called equilibrium constants, Ki values, etc. So the tighter it fits, the better. So here is our molecule, and here is what happens. So this piece is part of the enzyme, the heart of the enzyme. This is a serine. Actually, the enzyme itself is called a serine hydrolase. And in this case, I used a different molecule we invented. It's called AM6701. And I want to show you how this works. So this 6701, which is this molecule here, fits right here in that same. Okay. Then what happens then, this serine attacks it here, breaks this bond, and this piece now, this piece goes and is attached to the enzyme. This piece goes away. So the enzyme has come now, cut this molecule in two, and it got saddled with this piece. Now, because this molecule, this piece of the molecule is sitting in, this enzyme is unable to perform its function, it becomes inhibited. And there we have. Okay. So this is phase one. I'll give you a phase two. Now, enzymes are relatively easy to work with because they behave themselves. You can have the bugs, if you do it right, produce some of them. You can even crystallize them. But when we go to other proteins, like these here, protein structure, these are called receptors, it becomes more difficult to do it because these things don't sit well to crystallize. So you have to get the information piecemeal. And we had to devise ways of getting information. All we're after now is getting information because information is going to help us design new molecules. And again, the same idea, we have to make them fit. So we developed this method. It's called LAPS, ligand-assisted protein structure. And I want to show you how you go about collecting the information. Information doesn't always come in one package. They send you a letter. They say, well, these are the measures, and it's all done. You have to go and collect it and devise methods for collecting the information. So here's how we do this. In this case now, we start out with a drug. And we send this drug in, and we know that this drug goes and sits where it should be sitting because this drug is a potent drug. The question is, where is this drug sitting? We don't know anything about the structure of this big protein. So what we did is we made this drug, and we attach to it different hooks. So this molecule goes in, and when it gets into the place where it activates, it forms a bond. It hooks up with the protein. So we could then make this molecule, put a hook here, then we make it again, we put a hook here, we put it again, put a hook here. A hook is a group that undergoes what we call a covalent reaction. In other words, it goes in there and gets stuck. So think about it. If you can take that molecule, send it in, and then make it stick where it should stick, and that's the place where it is, and we can go back and find out where it is stuck, then we get information about where this molecule sits. And if we do it one at a time, we could find out one time it sticks here, one time there, one time there, then we have a contour of the molecule. We can go then and access all this information and use other methodologies to image it. So, for example, this molecule right here, it goes and forms a hook right there. This group here, when you put light on it, it covalently attaches here, or we can actually put two hooks on it, and one of them attaches here, and one of them attaches there. Okay? Then it becomes, the effort is in finding out where they are attached. And we do this by using some biology. We make mutants. Mutants is you take a protein, and you take one piece of the protein. In this case, we take a cysteine, and we transform the cysteine into a serine or something else. We do it again using molecular biology. And then by using this kind of combinations, we can find out where the drug sits. So we do it using mutational experiments, but we also do it through another method, which is a very important method called mass spectrometry. And mass spectrometry allows us to take the whole protein, chop it up, and find out which piece is attached where. Okay, anyone hear about mass spectrometry? Okay, all right. Well, mass spectrometry is a method which analyzes the structure of molecules and also quantitates molecules. You probably heard about, athletes being tested for drugs and finding out whether they have any drug. How do they do it? Mass spectrometry. Same kind of method. So mass spectrometry then is an additional tool which allows us to identify where these molecules fit. Then we take all this information together, and we pull it together, and we work with the computer, and now we know exactly how this molecule fits. It fits right there. See? The attachment is right here. Okay? And you see this molecule down. This is a helix, a helix, helix. So these are different helices that make up this particular receptor. This particular receptor belongs to a class of receptors called GPCR, G protein-coupled receptors. If you walk in a pharmacy and ask for a drug, about 50% chance that this drug works on a GPCR. So they are very good targets for making drugs. The beta blockers, they work on a GPCR. When you take a drug that is antihistamine, it works on a GPCR. If you make a drug be antidepressant, chances are it works on a GPCR. So we have now constructed this. We now have a way of knowing how these molecules fit. We could make new molecules to fit better, to fit in a different way. And you can play all kinds of games and scenarios here. You can make a molecule fit a little bit here and then touch it there, all kinds of things. But you have to go a little deeper in, because then you start having this conversation between yourself and the target. The target is a protein. And the more you talk to the protein, the more information you get, the more you know your way around, the better you can design these molecules. So I did want to leave you with these two examples. An example of an enzyme which we have cloned, and we are in the process of making what we hope will be good medications. We have one of these is being developed for Alzheimer's disease because, and we have a collaboration with our colleagues at Brigham and Women's Hospital. So this is one of the values of this thing. The other one is with the receptors. We are developing a drug for obesity. Control of obesity. It's a drug which we hope will work not only for obesity but also metabolic disorders, people who have fatty livers, people who are pre-diabetic, etc. They call CB1 antagonists. I have a picture to show you, which is very recent, and it's a picture from a piece of work that was done by a colleague, Brian Kobilka, and what they did is something technically very difficult. I mentioned to you that these receptors are difficult to crystallize. Well, Brian Kobilka crystallized that receptor. It's a different receptor. It's an opioid receptor. It's a receptor related to pain. And you see here how they got the crystals. And you see here the 7 transmembrane helices. And this is a drug called JD-TIC, not ours, someone else's. And you see it fits right here. They got this picture. And by the way, they did get the Nobel Prize this year. So this is about design and drugs. And you see these little entities there. They have shapes. They have personalities. And if you work a lot with them, you get to know about them. Thank you.
Get started. Well, first of all, thank you very much for sharing material with me, and I imagine with many of us, that is more unlike our comfort zone than anything we've had thus far. As I said, there are some architecture students in here, but mostly it's people from other colleges. So it's super informative for me personally just to see how this even, how this even starts. When I was putting together a list of people to visit this course, I thought something in this area would be interesting, but I really didn't know where to begin because I don't know anything about it. One of the first things, though, that I learned and was very much reinforced right here was how much based on shape and form this world really is, and it's not something I would have assumed. And your early diagrams were so clear about the heavier protein, I guess, and
right, and how it fits, yeah.
And how it fits in, and then on the second example of how sometimes you don't modify the initial receptor but the backside and so forth. That's very clear. There's a number of questions I'm going to have for you, but let's just start with one that was raised by your talk, and that is the need for getting tremendous specificity and about how these pieces fit into one another. We can have another conversation about how the explosion in digital visualization has clearly completely changed your world, I imagine.
Very much so.
There are obviously things that you could never have.
Sure. I mean, all these wonderful graphics, and graphics really have a language of their own. We used to have to draw all of these things.
I mean, just as people can't imagine how we drew some things by hand that they now see digitally, it's even more true in your world. But here's a question. Things mutate, both diseases, well, I guess the body is constantly in some sort of change. So is it the case that you're always looking for the most specific fit, or do you ever try to make a pretty close fit that would accommodate some change? Is that something?
Well, actually, it's a good question. Sometimes you may not want to have the drug fit perfectly. Because if it fits perfectly, it may not be able to get out. You want something to go in and out. So you have to allow for variations of this, and it depends on the context.
Right.
You may want to have a drug that goes in the enzyme and kills it. In other words, sits there forever.
Right.
Or you may want to have one that goes in and gets out easier.
Right.
And all of this depends again on the context. And all of these require special design.
Right. Right.
Now, regarding the mutations, it's an interesting story. What happens with the mutants is, and you've heard a lot about drug resistance.
Right.
And that happens especially in cases like cancer, or if you have an antibiotic or so.
Right.
So let's take the antibiotic. The antibiotic, the organism that is the deleterious organism gets hit by the antibiotic and gets killed. But then it learns, and then it changes its shape a little bit. Mm-hmm. And by changing its shape, this antibiotic doesn't work anymore, right, because it doesn't get in as well.
So that is an actual physical formal
Absolutely.
change.
What it does is it takes one of these amino acids, like I showed you how you can mutate. We mutated these amino acids. The bug does the same thing. It takes one or sometimes two amino acids, mutates them, and all of a sudden, this molecule doesn't fit anymore.
Right, right.
So now you have to counter it by making another one that fits in the mutant. So you design another one. And there are all kinds of strategies of how to approach this thing. The same happens with cancer. Mm-hmm. The cancer cells, again, learn about the particular drug, and then they mutate. So you have to design strategies which involve either making drugs that resist mutation, right, that resist the mutation ability of either the cancer cell or the organism, the deleterious organism, mm-hmm, or you may want to use two different ones, attack the culprit from two different directions, which is combination therapy, for example.
Right. Well, that's so interesting. I don't know about others, but that that puts a real face on this idea of, well, especially the thing we talk about most in the general press, I suppose, is growing resistance to antibiotics.
Right, absolutely. It's a very big problem today, because most antibiotics have developed resistant strains. Right. Even antibiotics for tuberculosis, right, right, malaria, right, and right, so on. So you have to make new drugs that go beyond that resistance, and they fit in the right places. So you can either go after the same target and make a drug now that can squeeze in because you've designed a new drug that can fit in, or you could say, okay, I'm not going to attack it here, I'm going to attack it somewhere else. Right. So I take another target, design another drug for the other target.
So the answer to the question is that no, you don't get more vague or looser to accommodate for the mutation. You simply have to redesign each time for the new situation, because you need a tight fit generally.
Yes. Yes. I mean, you could, I mean, there are, as I said, a lot of strategies. What you mentioned here, make it looser. Well, it's okay to be loose.
It's a carpentry model.
Exactly, exactly. I mean, it could be loose is fine as long as it works, mm-hmm, because you don't have to, if it's too loose, then it doesn't work. It mm-hmm comes off before it tweaks it. Right. So you have to play around with this. Right.
Well, this whole idea of there being a formal language underlying all of this is fascinating to me. So let me just get at some of these other questions. I think I've mentioned to you we've been using a set of questions or themes, if you want, that kind of tie together these different types of design, and I'll just very quickly. One of them is question, understanding the question, and I think we've alluded to that a bit, but we could get back to it. The context, empathy. Now this is perhaps less of an issue in your field, but we've spoken about the term empathy, particularly in the context of understanding a user, understanding the world from the perspective not of the person making the thing, right, but of the person who uses it.
I think you could extend it to the empathy domain, because you may make a drug that fits very well, but when it fits so tightly, it does things to the patient that are not very pleasant. Mm-hmm. So you may want to draw back and say, well, listen, I'm going to make one that's a little looser, right, so that it doesn't sit with you all the time, but it can metabolize. Right. So you have to accommodate the patient's needs. And actually, there's a whole process that goes beyond the initial design, because the initial design may fit very well. However, when it comes to the patient, there are all other parameters that have to be fulfilled, including the empathy towards the patient.
Sure.
So, and that would be drug development. It has to be, it has to reach the target. It has to sit there for a certain period of time.
Right.
You don't want a drug to sit there for a month.
Right.
You know, you want a drug that you take one pill every day, for example.
Right. So you have more control over
control.
The enterprise.
Absolutely. Yeah.
This is super interesting. So to continue with this list of words, we talk about iteration, which you've just in a way been describing, how there's a constant iterative battle because you're not dealing with a static foe.
Exactly.
With these diseases. And of course, in other design contexts, we mean iteration in the sense that often we have to make we have to make an attempt at solving an entire problem. If only, for example, Blake Middleton, the architect who visited with us, might make a sketch about the solution of an entire building complex, if only to have the ability to assess its strengths and weaknesses, and then circle back and do version 2 and 3 and 4.
But we have situations like this in the drug design area. We call them different generations. So first you start by making the first generation, and then you improve on that. So there are all these, so you have first generation, second generation. In my lab we call them first and second and third generation. And we make a lot of them. We don't make one.
Right.
We make a lot, and then a lot of them have to be really fitted almost by hand. In other words, you have to really put it in the tube there and see it work. Because it may fit in the computer, but there are things you may have overlooked. So you do have these iterations.
Your first look at these solutions is through computer modeling?
Well, there are different ways of going about it. In the old days, the drugs were all related to natural substances. The first drugs that were made were from natural substances. For example, aspirin was a byproduct of something called salicylic acid, or the drug atropine, which is made by nightshade, and they use it in the eye to constrict the pupil in the eye. Or you've heard, for example, that in the Amazon, the natives, they use these hallucinogens. Then, so what you do is you have a template there already. Or you heard that people who have a heart problem, they use this particular extract from a plant, digitalis. So what the chemists had done at the time is isolate these, and then use them. But then they said, well, maybe we can improve on them. So we can take this molecule there and tweak it. And so that would be a starting point.
Well, this is great, because that really suggests a kind of reverse engineering.
Exactly.
That the first thing, we find some things that work. We don't know why they work. This is even in the pre-scientific or pre-modern science. These things have alleviated various symptoms. And in the scientific era, we then try to break them down and see what are their constituent parts. And now, fast forward to the digital modeling world, where you can actually visualize why they work. You gave us I realize that this is a beginner's look at this thing, but the understanding of how the form, literally the shape of things. It seems to me that, as somebody who also lives in the visual world, I have my initial sense is that modeling can get better. But that's because of my ignorance probably. But these helical coils, I'm sure that is in fact the shape of things. However, I will say, as a designer, it's hard for me to understand as easily as you do that green business there fits in the coils. Like if you put that slide up and you said, George, for $100,000, Does that fit or not fit?
Yeah.
I would have to guess.
Okay. Now, now what is nice about the design,
Yeah.
and that's what I chose to use a generic way of representing these proteins, but there are other ways of representing, and the computers have been very helpful.
Right.
And then they can present them as solid pieces.
Yes.
And then you could almost see the whole and how it fits in.
Yeah, yeah. So it's more intuitive.
Yeah. So you can you can play around. You could really use the same type of information, and instead of putting it in the form of a coil, these are called alpha helices, for it. And these are very important building blocks in a lot of proteins. Linus Pauling was the first one to really find that particular structure. It's incorporated in a lot of different proteins. But you can get more detailed analysis. Like, for example, if you look at the drug there, that drug is done in solid form.
Right, right. We're talking about the bulbous green form.
Exactly.
Yeah.
You could do the same thing with the others. So you could have the solid form fit in a solid form.
Right, right, right. Well, this is interesting, because one of the things we haven't spoken that much about in this course, we've talked about visualization and notation some. And the times we have talked about it, it's been to very good end. For example, again, when Blake Middleton, the architect, was here, it was really useful, I think, to see how his very loose and imprecise early sketches, intentionally imprecise, allowed clients, stakeholders, to make the connection between a set of ideas and values and principles on the one hand,
Yes.
to the building blocks of a building.
Yes.
And if you got too much into the literal form of a building, there would be no way to make that connection. So you would have to do so in steps.
Yes.
And I imagine visualization in this is not dissimilar.
Actually, we work very similarly, except in this case it's not the client, but it's perhaps the colleagues, students, postdocs. We put together, maybe I'll draw something on the board, on a piece of paper. I'll say, okay, this is a nice structure to start with. What do you think? And then we say, okay, maybe we should really, and we have in mind what the other structure is. Maybe we could go this way. So it literally does involve incremental design.
Right, 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 right. Well, I think that's so important because, I've been reading the papers that the students have written for their first assignment, and they're generally very good assessments of why particular designs were successful. Oftentimes, I would say the vast majority of them have been commercial products, cars, iPads, things like this. But then they have to be clear about why the thing is successful, and you can imagine in almost every case that the final product was not drawn up right away. I mean, it's just that's not how things work.
Right.
That you have to work from what is the question, what exactly am I trying to solve here, to some incremental kind of building block steps until you find out what the real key issues to solve are, and then you can have focused attention on those, much as I'm sure once you find what the key links, connectors are, that's probably the tricky business.
Yes, yes, that's true. And of course we have the help here, as with architects, where they use nature as a source of inspiration. We have nature because nature is a very conserving, uses very conserving approaches for design. There's a lot of commonality, and nature doesn't go every time they have to make a new enzyme or a new protein, start from scratch. They have these pieces, and they put them together, and they have separate motifs, and they put them together, and it's pretty uncanny. And then the interesting part in the natural design is how you can make one target, one protein, differentiates itself from the other protein, because you don't want the two proteins to be the same. Well, otherwise they get mixed up.
That was one of my notes. What happens when...
Exactly. So they use building blocks, and then they tweak. The same way we tweak the molecules to make the drugs, nature tweaks its macromolecules to make it do specialized jobs.
To make sure that the same key does not open two different locks.
Exactly.
Yeah, that's interesting. So this, what you're suggesting about the sort of commonality of structures that one finds in nature in the architecture world, and in some other design worlds, we would call this typology, and that there are some, it's much simpler, but there's a typology to hotels, there's a typology to office buildings, there's a typology to row houses, to courtyard houses, and so forth. And you're right, you don't start from scratch and completely reinvent the wheel. There's a relationship between a series of essential components that exist in those building types, as presumably exists in the plant and natural world.
Yes, yes.
Yeah. Well, that's
did you say topology?
No, typology.
We use the term topology.
Yes.
Topology indicates the terrain of the molecule.
Yes, yes.
So you say this protein has a certain topology.
Now, is that the same use of the word that is found in mathematics?
Well, it's a little bit different than mathematics, and this is where the meaning doesn't exactly overlap with the mathematics.
Can I interrupt for 1 second? Topology and mathematics, and you guys probably, there's some math majors in here who know this certainly much better than I do, but the Mobius band and the Klein bottle and the complex kind of forms and surfaces is topology in mathematics.
Right, right. Here it has a general idea, but here it's a little more specialized, and it deals very specifically about the terrain. And topology comes from the Greek word topos. Topos means site.
Right. We would call that topography.
Exactly. It should have been called topography, but biologists like to use the word topology. Yeah, topography is a more accurate designation.
Right. And of course in our world there's a direct correlation.
Right.
There's actually a great book for those of you who want to know more about Boston called A Topographical History of Boston, which is a seminal book. Let me get back to our. Now this is interesting in your world because in some ways of the difficulty of visualization. I wonder if you guys ever use metaphor, if you ever use ideas to kind of.
Oh, absolutely, yeah. I mean, I always thought of molecules in a very anthropomorphic way.
Yeah.
Are they flexible? Do they dance? Do they do this and that? Are they very rigid? They don't move.
Right.
Absolutely, yeah. We anthropomorphize. I like to anthropomorphize molecules. It gives us a little more. Brings them a little closer to you.
Yeah.
And before the technology of visualizing these, where the technology was more limited, we had to dream these molecules up, how they show and how they do. Yeah.
Right. Well, that's super interesting. Now, there's a broad concept that's almost too broad to be useful maybe here, but the idea of synthesis. You're dealing with so many. To the layperson looking at these diagrams, there are so many parts. It's like looking at if you don't know anything about electronics and you look at an electronic circuit board, or if you look at the inside of a car, if you don't know anything about cars, you just think, I don't even know where to start. But I guess maybe metaphor addresses a little bit of what I was getting at with synthesis. In other words, how do you talk about these things as a whole? In other words, when you're dealing with a drug, I've got a high blood pressure. We're going to deal with that condition, but then you also have to take into account the whole enterprise.
Right. Well, nature does some very interesting things. You see, if you take an enzyme is a little machine. It's a biological machine, and the machine performs some duties. In this case that I showed you, what the machine does, it takes one molecule and cuts it in two. It just cuts. And there is a special place in the enzyme that does this. In this case, there were 3 amino acid residues, aspartate, serine, and histidine, and they get together, and the whole thing is they have one particular bullet, one point which attacks that bond and breaks it. So you could argue, why do we have to have the whole enzyme since the job is done by these 3 units together? And I've been doing some work on that. It turns out that the whole enzyme is built in such a way to optimize the job of these three. If you put these three together, they won't do it. But you need the whole enzyme architecture. And one of them pulls one way because these are electrons. You have to pull them one way, pull them the other way. And ultimately, this one warhead, which is the serine, becomes activated, and it does the job. So to come back to the proteins, the first thing we want to look at is where the action is. We call it the active site. And that's where we focus on. We start from there, then we go around. So not all aspects of that are equal. It's that part where the green molecule fits. That's the active site. And that's where we're after. Everything else is designed by nature to modulate the job of that active site.
This is fascinating to me because, you know, I'm used to seeing in almost any designed thing a whole, W-H-O-L-E, a whole that I can understand. Do you know what I mean?
Yes.
Whether it is almost anything.
Sure.
This is, in some ways, the most specific and particular example of design that we've had, but it's also the most unknowable as a whole for me, and I imagine for many people. So you know, the way that you, well, the way, let's zoom out a little bit, and the way that you arrive at questions. For example, have you been working on trying to solve one problem and stumbled upon another one altogether in this type of work?
Well, absolutely. I mean, this is the fun of the job, you know, because the most interesting things you find when you don't expect them, right? So yeah, that happens a lot, yeah. And I remember I had a graduate student years ago, and he'd come and say, well, you know, this didn't work, and he was very upset. I said, no, this is interesting because it didn't. Why didn't it work? You know,
right.
So you come up with things that you don't expect. But as far as shapes go, although there is a target site, the protein itself or the target, the DNA, they have their own shapes, and that gives you a different dimension to it. So both of these aspects are important. The site where the action is and how the whole entity modulates that particular activity, but also the shape of the whole protein, because that affects how these proteins behave and where they sit in the body.
You know, one of the things that we've talked about what constitutes a good design is that one of our working hypotheses is that a good design almost always solves more than one problem at a time. And I can see how, in this world, that would almost surely be true.
Absolutely.
That, yes, my preliminary objective is I need to cut these things in two, but then it also must meet some other criteria.
Sure.
And that's what resists the linear thinking.
Yes.
That I think sometimes people can come away from studying some courses or some disciplines with, which is not as, you know, I think one of the big lessons in studying and thinking self-consciously about design is that it is a tool for solving higher level problems, because it's not, if you're given, you know, we've talked about this before, but if you're given a single stage problem to which you need to just get, you know, the right answer on a math problem.
Yes.
That's not the highest level kind of problem you're going to be facing in life. In fact, most all of the hard ones are multivariable problems and require a toolkit that deals with multiple variables, solves more than one problem at a time. And I think that's, you asked me in my office before we came over here about this course, and that's really one of the things I think we're trying to get at. Is this is a way of dealing with higher level problems, ones where you're partly responsible for figuring out what the problem is.
Yes, and actually that's why these different activities and these different players allow themselves to be anthropomorphized, because we're pretty complex, but these are also very complex. So there's a kind of a communication level here.
Yeah. Well, let me. So I spoke with Murray Gibson, the dean of the College of Science, somewhat desperately the other day. I said, "I've got my first scientist coming. What am I going to? I don't know how to do this." He said, "Not to worry. Just tell him one thing: form equals function." And he will. And of course, unfortunately, you've already given us a bunch of cues about that, but it wasn't something that was obvious to me at the start. In other words, traditionally in the design world, in architecture, there's Louis Sullivan, the great 19th century architect, once famously said, "Form follows function." Yes. Well, the idea that form equals function is a real hint about what this whole enterprise is.
Sure.
About form making. And we've talked about what I was thinking of as the problem of mutations, and the answer is you have to keep remaking the fit. But I wonder if you could tell us just a little bit more. I mean, you went into some detail that is difficult for me to follow, so I wonder if we can kind of zoom out and talk a little bit about the two types of processes that you were talking about in your presentation. The first one, as I understood it, is the enzyme cloned approach that you're using on Alzheimer's. Is that right? And then the second one was about receptors.
Receptor, right.
And now,
similar to the picture I had at the end there, the crystal structure.
Yes. And receptors is what's being used on obesity.
Yes, in this particular case. Yeah.
And so receptors is about either blocking, is about blocking a bodily response.
Right.
So maybe you can just say a bit more about that, just because
sure.
I might be slower than the rest of our
right.
group on that.
Well, I think an easy example, because we all are acquainted with the high blood pressure.
Sure.
And everyone probably knows what a beta blocker is. A beta blocker is a drug you take when you have high blood pressure, etc. It works through a receptor. It's called the beta adrenergic receptor. Mm. And what it does is, when you get the right drug, it prevents the native hormone, which in this case is called norepinephrine, for acting there. So instead of norepinephrine going in there, you got the drug, and then norepinephrine cannot do its job.
And the norepinephrine is in my body.
In your body.
And so we don't like the effect that it is creating.
Yes, because it went a little haywire, so much so that your vessels now have become constricted. And you have high blood pressure.
Causes my heart to work more.
Causes your heart to work more. Some people eat a lot of, let's say, fat and so on. The vessels, the blood vessels get clogged. If they get clogged, then the blood pressure becomes higher, right? When you have a, so what you want to do is you want to relax them a little bit.
Right.
So by blocking that, because what norepinephrine does is constricts the blood vessels.
Right.
So by blocking its effect, it allows the blood vessels to relax. So that's the type of, and the way it gets from the drug to get to the blood vessels is not an easy path. It involves many, many steps. The ultimate step is where your blood vessel now relaxes. But it goes through a process. There's something called the G protein, which couples with this, and it goes to other proteins which are called kinases, and all of that ends up in that physiological effect.
What was was there a particular breakthrough in between the sort of reverse engineering approach to drug discovery to this receptors category? Was there a particular breakthrough in fairly recent history that led to this area of drug development?
Well, the receptors came into play much before they were known, because we've been using drugs targeting the receptors from ancient times.
But without knowing it?
Without knowing it.
I see.
Actually, when I went to school, we didn't know anything about receptors. And I remember my professor, I said, what is a receptor? He says, So receptor is like God, you know it's there, but you can't see it. Well, it's been now about, so the major breakthrough happened when we were able to clone things. There was a period of time when some very large laboratories in the world, they were trying to isolate these receptors. And they were focusing on one particular, which is the cholinergic receptor. And that happened, they had these fish, eels, electric eels. And the electric eel has a whole machinery there which produces electricity. And it's done through chemical means. And it's done through that receptor. So they were able then to get amounts of that receptor, trying to purify it and find out what the structure is, etc. But that's way after we were using these molecules, etc. However, what happened is, while all these laboratories were trying to get, cloning came in. In other words, you could use the prototypical information piece, which is the DNA, and produce these proteins from scratch, having the bugs produce them. That then opened the door to doing a lot of discoveries.
I see. So you could take a sample of a part of a body and then use it as the basis, as the base,
sure, and have it cloned. So that's how they cloned all of these receptors. So these big labs lost their great value at the time. They never got a Nobel Prize on that.
And how long ago did this happen?
Well, this is it's not really that far. It's maybe about 40 years ago.
Wow.
Yeah, it's a big revolution in.
Well, I know there has been. Huge revolution in drug
yes.
efficacy and the sorts of things that you can actually treat now.
Right.
We see it, of course. One of the consequences is we see on television advertisements directly to patients.
Yes,
for
that's
all
something.
These things,
Yeah.
I always think it's somewhat questionable. I'm supposed to go argue with my doctor.
About
what.
Yeah, I agree with you. Actually, most scientists in industry agree with you. Yeah.
Well, let me ask the audience if we have any questions for Dr. Makryannis about this fascinating world of drug discovery. Yes.
How do you even begin to visualize all this stuff before you have
Yeah. Actually, it's a good question, and people think of science as being an activity where you wake up one day and you have it, and you see the light. But it's actually a very incremental, and all of science is a very incremental activity. So you start and you keep building and building. Think of it as a camera, and you have a field of vision, and the first time you look at it, it's pretty opaque. You can't tell. And then you do more and more work, then you get to see it clearer and clearer. So the answer is work. You just have to do a lot of work.
Just in the last, if we say that you've been, the drug discovery really exploded over the last 40 years, in the last 10 or fewer years, the level of digital visualization has just exploded.
Well, it's been very helpful. I can't say that the technology, the results that were obtained because of the new technology have been commensurate with the advances in the new technology.
Okay.
It's still a difficult process because, as you mentioned, it's a multifactorial.
Right.
And there's a lot of things that involve different disciplines, different details. Right. For example, if you look at cancer, we've made some. There's been some improvements, but the cancer drugs that are available today, you can count them in two hands.
Yeah.
So it's been a difficult process because it involves different activities, and scientists in general like to focus on one activity. What's interesting, what I find interesting in drug discovery, is that you have to bring in a lot of different components that are not necessarily congruent. And one difficulty, for example, with drugs, which you may not pay a lot of attention to, you could make the perfect drug, but then how do you get it where it should go?
Right.
That's so there are a lot of things. So I would say there's been a lot of good advances in drug research because now the FDA requires that you must know how the drug works before you give it.
Mm-hmm.
So it does require you to know the target. It does require you to do all of these. So the drugs we make today are safer than they were before. They're not as plentiful.
Right.
Well,
the regulatory environment has grown right along with the industry, I'm sure. So
sure.
the public interest can be
Sure.
modified that way. There was another question over here.
Yes,
yeah,
sir.
Here, just a second. Let me give you this.
In your lab, since you are doing drug discovery, do you have like different people that like major in bio and chem, or do you just have like people that have taken specific courses like biochem and stuff?
Sure. So we are a multidisciplinary laboratory, which doesn't mean that everyone does everything. So there's a group of people who do the so-called cooking, organic synthesis. Once you decide what you're going to make, they go there and make it. And these get trained in organic synthesis. There's a group that looks at the structures of these proteins. We use NMR or computation. It's called biophysics. There's a group that looks at how these molecules interact with the cell. What's the biochemical and what kind of measurements you make. How do you know that this thing sticks in there? You have to make some measurements. So my lab is divided in three different, one of them is called chemistry, and they also know a lot about computers, etc. But they do a lot of synthetic organic chemistry. There's a group that does cloning and mutations and all of this. We call them biochemistry. And there's a group that does the biophysics, which is the measurements like the NMR, etc. And they can be from different departments. So I have students who are from the chemistry department, students from pharmaceutical sciences, students from biology. And actually we have one student from engineering.
So all these groups are informed about everything?
Yes. What is nice about it, although if you're a student you don't end up doing all of these things, but we all get together and we see how they mix. So the contribution there is to see how you can bring the disciplines together. And although you may not know how to do the actual cloning, you know what it means and what the context is. So you become multidisciplinary.
Other questions?
Hi, thank you. I'm just interested, I mean, we all see in commercials, if you take this drug, these are the side effects, and the only thing they don't tell you is you might even die using it.
Yeah, sometimes.
So I'm just wondering if that's one of the effects that you said that the drugs can probably fit into more different places.
Right, exactly.
Is that
That's
why?
it.
Is that the only reason why, or?
Well, I mean, you see there is, a drug always has potentially toxic effects. In fact, in the old days, to test whether a drug has any efficacy at all, you give high doses to an animal, and if it doesn't die, then it's no good. You know what I mean? It's called the LD50. It's called the lethal dose 50. In other words, what is the dose that would kill half of the? We don't use this anymore, and it's not used there. But a lot of the toxicity is because of side effects. That's why you want to get it in the right place. And however, the full story is more complicated because sometimes you may need to synergize. If you get two things to happen at the same time, it can give you a better result than one thing by itself. But a lot of the toxicity effects are because they go to other places.
Other questions? Well, Professor Makryannis, I want to thank you very much for joining us.
Well, thank you very much.
Well, thank you. Really, it was wonderful.
Yeah.
I'm so glad you came.